This is the first video in the series about addictions. The goal of this video is to define and classify drug addictions, behavioral addictions, and also discuss the normal physiology of the reward system. We'll see what's the difference between feeling pleasure and excitement versus feeling boredom, not superficially but on the cellular and molecular levels. Let's start. So, what is addiction? Addiction is a biopsychosocial disorder characterized by compulsive engagement in rewarding stimuli despite the adverse consequences. Compulsive means the person has an insatiable urge to engage in rewarding stimuli. The word biopsychosocial means addictions have interconnections with biology, psychology, and socio-environmental factors. Addiction affects the reward system in the brain. The reward system is responsible for desire or craving for a reward, associative learning, and positively valenced emotions, particularly ones involving pleasure as a core component. For example, joy, euphoria, and ecstasy, which is an overwhelming feeling of great happiness or joyful excitement. A rewarding stimulus is any stimulus, object, activity, or situation that has the potential to make us approach and consume it. According to the rewarding stimulus, addictions are classified as drug addictions or substance addictions and behavioral addictions. Examples of substances that can cause addiction are heroin, cocaine, alcohol, nicotine, methamphetamine, opioids, and amphetamine. Behaviors identified as being addictive include gambling, sexual intercourse or use of pornography, use of computers, playing video games, use of the internet and other digital media, exercise, food, and shopping, or how some people call it retail therapy, which is ironic because it's not therapy. It's important for every person to know about addictions, because they can affect our behavior, motivation, health, and our life in general. Notice I didn't say drug addictions. I said addictions in general can affect our lives. Much more people have behavioral addictions than drug addictions. Although this sounds kinda abstract now, by watching this series you'll understand. This is quite serious. Our everyday lives revolve around rewarding stimuli. Before we understand how different types of addictions affect our reward pathway, we need to understand the normal physiology of the reward system. Here you can see the medial view of the brain. Three main structures of the reward system that you must know are the ventral tegmental area, or VTA for short, which is situated in the midbrain, adjacent to the substantia nigra, the nucleus accumbens, and the prefrontal cortex. In order to give you some spatial orientation, let's take a look at the cross-section of the midbrain. This is the anterior side of the midbrain and the posterior side, respectively. There are three main parts of the midbrain. The tectum, which consists primarily of the superior and inferior colliculi, the crus cerebri, which contains the motor tracts, traveling from the cerebral cortex to the pons and the spine, and the tegmentum. The tegmentum is everything between the tectum and the crus cerebri. Here you can see the substantia nigra. The ventral tegmental area is located medially and a little bit posteriorly to the substantia nigra. Engaging in enjoyable activities generates action potentials in the dopamine-producing neurons of the ventral tegmental area. But how does the VTA know about your activities or emotions? There must be a connection between different parts of the brain and the VTA, right? Of course there is a connection. In our example, the boy is eating. What things could stimulate the VTA in this case? Think about this. When you're eating, the main three things that you're enjoying are the taste of food, the sight of food, and the smell of food. The parts of the brain responsible for sight, taste, and smell, respectively, will stimulate the VTA. Of course, it's a little more complicated than that. It always is. I intentionally omitted some neurons to create a simplified version of what's happening. But when we discuss behavioral addictions, you'll see all these omitted neurons and you'll be crying, I promise. The connections between different parts of the brain and the VTA are essential in understanding behavioral addictions, like food addiction, for example. That's why we'll discuss them in more detail in the upcoming videos. The ventral tegmental area contains a lot of neurons that produce and release dopamine, called dopaminergic neurons. These neurons will send axons to the nucleus accumbens, which is a component of the limbic system, and to the prefrontal cortex. The pathway that connects the ventral tegmental area to the prefrontal cortex is called the mesocortical pathway. The pathway that connects the ventral tegmental area to the nucleus accumbens is called the mesolimbic pathway, also known as the reward pathway. In a healthy person, the reward system reinforces important behaviors that are essential for survival, such as eating, water drinking, sex, and social interaction. For example, the reward system ensures that you seek food when you are hungry, because you know that after eating you will feel happy and rewarded. In other words, it makes the activity of eating pleasurable and memorable, so you would want to do it repeatedly whenever you're hungry or hangry. Okay, now let's study some details about the nucleus accumbens and the prefrontal cortex. The nucleus accumbens is one output of the VTA projections. The nucleus accumbens itself consists mainly of the medium spiny neurons, which are GABAergic neurons. GABAergic means these neurons produce gamma-aminobutyric acid, or GABA for short, which is the main inhibitory neurotransmitter in the mammalian central nervous system. Let's zoom in on this synapse in the nucleus accumbens. Here you can see the dopaminergic neurons from the VTA that form the mesolimbic pathway. The components of these neurons are the cell body, axon, the synaptic end bulb, where we can find vesicles filled with dopamine, and you can also see the dendrites of the GABAergic medium spiny neurons from the nucleus accumbens. When an action potential arrives from the VTA, dopamine is released from the neurons into the synaptic space and binds to dopamine receptors located on the medium spiny neurons and stimulates them. On the postsynaptic membrane are 5 types of dopamine receptors, named dopamine receptor D1, also known as DRD1, and just keep changing the number until you reach dopamine receptor D5. I'll represent the 5 types of receptors in different colors. Dopamine receptors D1 and D5 are called D1-like receptors. They are coupled to the G-stimulatory protein and mediate excitatory neurotransmission, meaning they stimulate the neuron. Dopamine receptors D2, D3, and D4 are called D2-like receptors. They are coupled to the G-inhibitory protein and mediate inhibitory neurotransmission, meaning they inhibit the neuron. Neurons in the nucleus accumbens are mostly the medium spiny neurons, containing mainly D1-like receptors or D2-like receptors. Approximately 40% of the medium spiny neurons express both D1-like receptors and D2-like receptors. The D2-like receptors are the most important in understanding the physiology of the reward pathway, and you'll see why in just a second. In a person with addictions, some molecular and cellular alterations take place inside the medium spiny neurons of the nucleus accumbens and other neurons. The nucleus accumbens is associated with acquiring and eliciting conditioned behaviors. It plays an important role in processing the rewarding stimuli. But how exactly does this processing take place? I'll explain using as an example food addiction. Please remember one thing. The predominant response of the medium spiny neurons in the nucleus accumbens to rewarding stimuli, such as food for example, is inhibition. In other words, reducing the excitation of the neurons in the nucleus accumbens is rewarding. Okay, that's a little bit confusing, right? Inhibition equals pleasure? How so? Don't worry, it makes so much sense and it's really easy. Quite easy. Almost easy. Let's take a closer look at one dopaminergic neuron from the VTA. You'll be able to see the cell body, the axon, the synaptic end bulb which contains vesicles filled with dopamine, the mesolimbic pathway formed by these neurons, and one GABAergic neuron from the nucleus accumbens with its dendrite, cell body, axon, and the synaptic end bulb which contains vesicles filled with GABA, the main inhibitory neurotransmitter in the CNS. Now imagine you're eating your favorite food. The parts of the brain responsible for sight, taste, and smell will stimulate the amygdala, which stimulates other neurons, which in turn will stimulate the dopaminergic neurons from the VTA. The dopaminergic neurons will release dopamine, which will bind to the receptors on the medium spiny neurons and will inhibit them. But how? The inhibition happens due to the D2-like receptors. They are coupled to the G-inhibitory protein, which will inhibit the neuron. Okay, so now the GABAergic neurons are inhibited. Because of that, they will release little to no GABA. Normally, GABA binds to GABA-A receptors. Chloride ions enter the neuron and hyperpolarize it, thus inhibiting it. But since there's little to no GABA in the synaptic cleft, very few chloride ions are gonna enter the cell. If GABA doesn't inhibit the neurons from the parts of the brain responsible for pleasure anymore, this is called disinhibition, or you could say stimulation. This stimulation creates the experience of pleasure. So what happens when you're doing something that is qualified as boring? Let's say right now you're stuck in traffic. Your visual and auditory stimuli from the traffic will reach the visual cortex and the auditory cortex. But since this information is not so necessary for survival or evolution, it won't stimulate the amygdala so much. In other words, it will inhibit the neurons in the amygdala. As a result, the amygdala won't stimulate the neurons connected to it. Those neurons won't stimulate the dopaminergic neurons of the VTA so much. The dopaminergic neurons will release little to no dopamine. Dopamine will no longer inhibit the medium spiny neurons. This is called disinhibition, or you could say stimulation. The GABAergic neurons will release a lot of GABA. GABA will bind to GABA-A receptors. Chloride ions will enter the neuron and hyperpolarize it, thus inhibiting it. In this way, GABA will inhibit the neurons from the parts of the brain responsible for pleasure. This inhibition will create something that's opposite to the experience of pleasure, which is displeasure, unhappiness, and boredom. This is the essence of the whole process. Now, one more thing. Do you remember I said parts of the brain responsible for pleasure? We already know that the red neuron in this picture is responsible for pleasure. Now we have to see where in the brain is this red neuron actually situated. In order to do that, firstly, let's take a look at the coronal plane of the brain. Here you can see the putamen, the dorsal pallidum, which is commonly known as the globus pallidus, and consists of the external and internal segments. Then we have the ventral pallidum, the thalamus, and the prefrontal cortex. The dorsal and the ventral pallidum combined form the pallidum, which is a part of the basal ganglia of the brain. And here again you can see the pallidum. In order to create some space, I'll make the coronal section of the brain smaller and add the VTA and the nucleus accumbens to the picture. Another very important term when it comes to the study of the reward system and addiction is the striatum. The striatum is composed of 3 nuclei. The caudate nucleus, you can see it in red. Then we have the putamen and the ventral striatum. The ventral striatum contains the nucleus accumbens. The internal capsule separates the caudate nucleus and the thalamus from the putamen and the globus pallidus. This coronal section was taken right here. Now let's take another coronal section more anteriorly. Why more anteriorly? Because only there we'll be able to see the nucleus accumbens. On the left side of the screen, you can see the lateral view of the brain. The green line represents the new coronal section of the brain, and the blue line represents the old coronal section. Now let's have a quick look at the horizontal section to avoid any confusion. The old coronal section was made right here. We were able to see the putamen, globus pallidus, thalamus, the internal capsule, and the caudate nucleus which runs above. Also, in the left corner, you can see the parts of the caudate nucleus, which are the head, the body, the knee or gentle, and the tail. So, the new coronal section will be taken more anteriorly, right here. In the anterior coronal section, you'll be able to see only the caudate nucleus, the putamen, and the internal capsule between them. This is how the anterior coronal section looks like. Again, the striatum is composed of 3 nuclei, the caudate nucleus, the putamen, and the ventral striatum. The ventral striatum contains the nucleus accumbens. The internal capsule separates the caudate nucleus from the putamen. But why is it called striatum? I'll explain. Striatum in Latin means striped. The white matter of the internal capsule, overlaid with these gray matter bridges, creates a striped appearance. That's why this area is called the striatum. It's striped like a tiger, or the American flag, right? Now let's go back to the old cross-section of the brain. We are interested to see which parts of the brain are responsible for pleasure. Let's see where in the brain is this red neuron actually situated. We'll start with the medium spiny neurons. The medium spiny neurons of the nucleus accumbens will send axonal projections to the basal ganglia and the ventral analog of the globus pallidus, known as the ventral pallidum, which is a major center of reward in the brain. The ventral pallidum, in turn, projects to the medial dorsal nucleus of the dorsal thalamus, which sends neurons to the prefrontal cortex. The neuron in black is one medium spiny neuron from the nucleus accumbens. When this neuron is inhibited, it releases little to no GABA. As a result, the neurons in red, which are responsible for pleasure, are stimulated and they release glutamate. Glutamate serves as the major excitatory neurotransmitter in the vertebrate's central nervous system, and also is the main neurotransmitter of communication between the thalamus and the cerebral cortex. So, one more time, those 2 neurons in red are responsible for pleasure. Notice that in the end both the mesolimbic pathway and the mesocortical pathway send their neurons to the prefrontal cortex. The prefrontal cortex plays a central role in cognitive control functions, such as adaptive thinking, planning, self-monitoring, self-control, our working memory, also known as short-term memory, time management, and organization, as well as in our reasoning, judgment, decision-making, behaviors, and even our personality. Now let's discuss the functions that are specific to those 2 neurons in red. We already know they can induce pleasure, but that's not their only function. That red neuron in the prefrontal cortex can also modulate cognitive control functions, thereby influencing attention, impulse inhibition, prospective memory, which is defined as the memory for intentions, and cognitive flexibility. Think about this example. When you're really enjoying your favorite food, the pleasure can capture your attention. You feel good at the moment and you'll remember the taste of that food and the pleasure associated with that specific taste. Once again, to avoid any confusion, what is pleasure? Pleasure is that good feeling you get in response to rewarding stimuli, such as food and addictive drugs, for example. If we're talking about appearances, a person that experiences pleasure might be smiling or crying from happiness maybe. But if you're wondering what happens inside the brain while feeling pleasure, the answer lies in those red neurons. Roughly speaking, if the neurons in red and their connections are stimulated, you will experience pleasure. Okay, now back to our example. We've discussed that the pleasure can capture your attention. Also, you'll remember the taste of that food and the pleasure from it. What else? In the future, when you'll encounter your favorite food again, you already know that consuming it is pleasurable because of the memories. The question is, will you eat it or will you restrain yourself? See? Impulse inhibition. Impulse inhibition is the ability to control and inhibit our desire to act according to some of our impulses. An impulse is a sudden wish or desire. In our example, the impulse or the desire is to eat all that tasty food from the plate. But you can restrain yourself through impulse inhibition, which is modulated by that neuron in red from the prefrontal cortex and its connections. So again, attention, memory, and impulse inhibition are all modulated by this neuron in the prefrontal cortex. Speaking about addictions and memory, there is a connection between the prefrontal cortex and the hippocampus. Do you see these dark blue neurons? These neurons are particularly important for memorizing details about the rewarding stimuli. Because of the connection between the prefrontal cortex and the hippocampus, an addict can form associations between the memory of the rewarding experience of drug use and the memory of the cues in the environment. For example, the aspect of the room where he took his last dose, or the aspect and mood of his drinking buddies. As it turns out, the cues in the environment are strong mediators of the drug-seeking behavior and can trigger relapse even after months or years of abstinence, just like in this scene from BoJack Horseman.
Shark fin and white rhino horn. Sober, so good. Whew. BoJack?
Hey, Sarah Lynn. You want a party?
Oh, thank God. Yes!
Okay, for those of you who are still confused, in this scene, Sarah Lynn is the addict, alcohol is the rewarding stimulus, BoJack represents the cue in the environment because he's Sarah Lynn's old drinking buddy. This cue in the environment is responsible for triggering alcohol-seeking behavior and relapse after 9 months of abstinence. Maybe this example is a little bit exaggerated, but that's good, because you'll remember it better. So you see, it's very easy to relapse. Studies reflect that about 40 to 60% of individuals relapse within 30 days of leaving an inpatient drug and alcohol treatment center, and up to 85% relapse within the first year. Different addictive drugs like heroin, cocaine, alcohol, nicotine, methamphetamine, opioids, and amphetamine have different mechanisms of action, but the common outcome is that dopamine builds up in the synaptic cleft to a much greater amount than normal. Here you can see the difference in dopamine concentration in a normal individual versus the concentration in a drug user. See? Big difference. We can observe the same buildup of dopamine while experiencing addictive behaviors like gambling, sexual intercourse or use of pornography, use of computers, playing video games, use of the internet and other digital media, exercise, food, and shopping. Here you can see the difference in dopamine concentration in a normal individual versus the concentration in one with behavioral addictions. Here you can compare these three cases. The highest concentration of dopamine will be in drug users. The buildup of dopamine in the synaptic cleft, especially due to drug usage, causes a continuous inhibition of the medium spiny neurons from the nucleus accumbens and is responsible for prolonged and intense euphoria. High concentrations of dopamine in the synaptic cleft are dangerous because they can induce addiction-related structural changes in the medium spiny neurons of the nucleus accumbens. We'll discuss these changes in one of the next videos. I think that's enough information for this lecture. This was the introductory part. Alright guys, thank you for watching. In the next video, we'll study more about the behavior in addictions, so stay tuned. Good luck!